In this lesson, we'll be talking about smear preparation and simple staining. So in our prior microscopy lesson, we talked about how bacteria naturally don't have pigmentation. So if you tried to put them against the background of a clear microscope slide, you would not be able to create that contrast to see them clearly. So in order to achieve contrast, what we need to do is we need to stain the bacteria using chemicals called dyes. That will stain them some sort of color depending on what type of dye you're using, but for example, purple, blue, green, pink. And that color contrast against a clear microscope slide is going to allow us to be able to see them clearly. So one type of dye that we have is a basic dye. Basic dyes are used when your intention is to stain the cells. Basic dyes have a positive charge, and bacterial cells have a negative charge, and they have a negative charge because of the phospholipids in their cell membrane. So we know from prior classes, opposite charges are going to attract to each other. So the positively charged dye is going to be attracted to the negatively charged bacterial cell. The two will bind, and then that is going to allow the bacterial cell to turn some type of color. So if you're using something like methylene blue, obviously the cell would stain blue. Crystal violet, it would stain purple. Malachite green, green, and then safranin is like a reddish pinkish color. But it is important to remember with basic dyes, your intent is to stain the cell, and it does require to use a dye that has a positive charge, which we refer to as a basic dye, because that allows the dye to bind to the negatively charged cell and give it that color and create the contrast so that you can see the specimen under the microscope. We use basic dyes typically when we want to do a simple stain, and simple stain is kind of exactly what its title refers to. It is simple in the sense that it only requires the use of one single dye, because as we advance, we'll eventually get to the point where we do differential staining, which requires two or more dyes. But we typically start out with simple staining, which just requires one dye, but that dye does need to be a basic dye because it needs to have the ability to bind to the bacterial cells and give them color to create that contrast. So when we're doing a simple stain, we do use that one basic dye, and our intention is to look at just general characteristics. Using a simple stain, you're not going to learn very specific details about a bacteria. So for example, the thickness of the cell wall, or if it forms endospores or anything like that, but it's just going to show you those kind of general physical characteristics, which are size, shape, and arrangement. So this chart goes over some of the different shapes and arrangements that bacteria can make. Definitely the two most common that we'll see in our lab and in our course are the cocci bacteria and the bacilli bacteria. So cocci bacteria are going to be circular in shape, whereas bacilli are going to be rod in shape. Now with cocci, you can have just a single coccus, but with cocci, a lot of times when they divide, they'll stay attached to one another and form all these unique arrangements. Cocci definitely have the most diversity as far as forming arrangements. So you can have things like diplococci, which is a pair of two, staphylococci, which is an irregular cluster, like a grape-like cluster, tetrads, which are basically a square formation of four, streptococci, which are chains of cocci, and then Sarkinia, which is like a 3D cube of circular bacteria. But cocci is definitely a common genus that we look at, or common shape that we look at. Bacilli is another very common shape that we look at. So with bacilli bacteria, they typically need to form some type of linear arrangement, so they're not going to do anything like staphylo or tetrad. They need to be arranged in some type of line. So you can just have a single bacillus, which we sometimes refer to as like a monobacillus. We can have diplobacilli. We can have palisades, which are stacked in this type of arrangement together. And then streptobacilli, which is a chain of bacilli. There are strains that form, or species that form coccobacillus, which is kind of a hybrid between cocci and bacillus. So kind of tend to refer to that as just a very short rod. That's common for something like E. coli. There are also some more rare shapes over here. So for example, Vibrios are curved rods. We have spirochetes, which kind of have this spiral type appearance. But definitely in this course, what you're going to become most familiar with are the cocci, the circular bacteria, and the bacilli, which are the rod bacteria. Now, to do any type of stain, really, the first thing that you have to do is create a smear. So by smear, I mean that you have to add the specimen, in our case bacteria, to the slide so that you can then go through the staining procedure. So when we want to create a bacterial smear, what we typically do is we take a loop full of water and add it to the microscope slide. And then into that loop full of water, we add a small sample of bacteria. And again, to get that small sample of bacteria, you would have to sterilize your inoculating loop, collect a small sample, and then take that small sample of bacteria and basically smear it back and forth in a drop of water. If you're pulling a sample of bacteria from like a nutrient broth that's already a liquid, you don't need the drop of water. You can just take the loop full of broth with bacteria and just smear it on the slide. But a lot of times in our course, we're pulling bacteria from a petri dish, from a plate. So for us, we have to add the drop of water first and then add the sample of bacteria into that drop of water and just smear it across. Once you've smeared the bacteria throughout that drop of water, the next part is just a waiting game. You basically have to wait until the slide is completely dry. And you'll know when it's completely dry because you'll get this kind of cloudy haze that runs across the slide, and that cloudy haze is from the bacteria. You do not want to go through heat fixation when there's still moisture on the slide because it'll essentially boil the bacteria and they'll burst and explode, and then you'll just see bits of bacteria and not the actual whole cells. So it is really important to let that drop of water with the bacteria that's been smeared out into it, to let that air dry before you move on to the next step, which is heat fixation. So heat fixation is basically just applying heat to the microscope slide that now contains the bacteria. Heat can be applied in a bunch of different ways. So this example shows the slide being passed through the flame of a Bunsen burner. That's one way to heat fix it. You could also set the slide on the hot plate for 30 seconds to heat fix it. You could even technically hold a lighter underneath it to heat fix it, which is I've seen used in the industry at like my vet's office. So really it's just a term for applying heat to the slide. In our course, we tend to favor Bunsen burners or the hot plate. So what heat fixation is going to do is going to, one, adhere the specimen to the slide. We want the bacteria to stick to the slide because when we go through the staining process, we're going to be rinsing the slide with water and various different dyes. If it's not stuck to the slide, then you're going to lose it in the rinsing into your staining trays. The next thing it does is kills the specimen. So we do want to make sure that the specimen is killed, stuck to the slide, obviously for biosafety concerns. And then the last thing is it weakens the membranes for stain retention. Stains to bacteria would be considered toxic chemicals that they would not want to let in through their membranes, but by heat fixing them, it kind of breaks down that cell membrane a little bit so that the stain can be easily absorbed in, and then we can get that contrast to see the specimen under the microscope. So this kind of just walks through the various steps I just talked about. So we do need to kind of create that smear and spread it by placing the bacteria in the drop of water and smearing across that slide. Then we're going to completely let it air dry, get that cloudy haze, and then we're going to heat fix it, which basically is applying heat, whether that's through a hot plate or through the use of a Bunsen burner. Once you've heat fixed it and let it cool down for a minute or two, then you're ready to go through the staining process. So with a simple stain, it's very easy. All you have to do is add a few drops of any type of basic dye. So for example, crystal violet. You just add a few drops of crystal violet onto the slide and let it set for a minute or two. Every type of dye has like a time limit of how long it needs to set. Most of them are about a minute or two. But you would let it just set for a minute or two so the bacteria will absorb the stain, and then you just rinse it off with water, and then you look at it under the microscope all the way up through oil immersion, and you'll see something like this. So this is Staphylococcus that's been simple stained with crystal violet. So all the cells are purple, but we definitely see that size, shape, and arrangement. So we see that cockeye shape, and then with this we can see that most of the cells are in that staphylo or irregular cluster type of arrangement. Now there's definitely some mistakes that can happen along the way. One of the mistakes is making your prep too thin, which is basically not adding enough bacterial cells. So when you collect your sample from the Petri dish, you want to make sure that you collect from a region of the dish that actually has a sample of bacteria there and not just like a blank part of the Petri dish. Because obviously you want a good sized sample to be able to see the cells once you stain them and put them under the microscope. This one's not super common. What is definitely more common is making the prep too thick, which means adding way too much bacteria. In microbiology, less is more. So when you go to collect a sample of bacteria, you may not see anything on your inoculating loop. And that's perfectly fine. They're microscopic. There's probably thousands and thousands and thousands of them there. But a lot of times students get concerned that they don't have enough, so they take a big old scoop of bacteria from the Petri dish and then mix it in that, or smear it in that drop of water on the slide. And that adds way too much bacteria. And what you'll see under the microscope is just a big blob of purple, where all the bacteria just kind of got clumped up together. And when that happens, you're not able to see individual shapes and arrangements. Another mistake is stirring with your inoculating loop. So when you take your loop that has bacteria on it and mix it into the drop of water on the slide, you want to smear it very gently. You do not want to aggressively stir it. The reason why you don't want to aggressively stir it is because you can break up the natural arrangement of the bacteria. So with this bacteria, Staphylococcus, it's supposed to be in irregular clusters, these grape-like clusters. If you take a sample of this and you stir it aggressively into that drop of water, you're going to break up those clusters and they're just going to have that single cell arrangement, which isn't what they naturally look like in the real environment. So we gently just kind of smear it to kind of maintain that arrangement, which is especially important when you're talking about things like Staphylococcus or Streptococcus that have those kind of unique arrangements. Another thing is staining too much or too little, which really doesn't have anything to do with quantity of stain, it's more about the time limit. If a stain requires you to let it sit on a slide for 1 minute and you rinse it off after 30 seconds, obviously not enough stain would have absorbed into the cells and then you wouldn't be able to see them clearly under the microscope because of lack of contrast. However, if you let the stain sit on for too long, so say we let it sit on for 10 minutes versus the 1 minute that it would typically need, what can happen is the stain can start to dry up and harden around the cell and form additional layers around the cell so it doesn't kind of rinse off. And it can cause the cell to appear much bigger under the microscope than what it should be naturally. And that's because the stain has kind of dried up and formed additional layers around the bacterial cells. So then you don't get that natural representation of how small bacterial cells actually are.